Welcome everyone to today's speaker series. Thank you all for joining. My name is Rosa Stalteri, and I'm the Knowledge Translation Manager with CanCOVID and the speaker series host and moderator. As new strains of SARS-CoV-2 are rapidly spreading globally, including in Canada, a number of questions are being raised, and scientists are racing to understand its implications. It is a pleasure to welcome Dr. Charu Kaushic, who will be sharing with us the latest developments on the emerging SARS-CoV-2 variants and the clinical, vaccine, and public health implications. Dr. Kaushic is a professor at the Immunology Research Center at McMaster University. She is a member of the Canadian Immunity Task Force and the scientific director of CIHR's Institute of Infection and Immunity. So before I hand over the floor to Dr. Kaushic, just a few little items to note. We welcome you to keep your camera on, but please do keep your microphones on mute during the presentation. And for the 10-minute question and answer period, we welcome you to raise your hand, or you can also use the chat box. So without further ado, I will hand over the floor to Dr. Kaushic.
Thanks very much, Rosa. So it's a pleasure to provide an update on the SARS-CoV-2 variants of concern and emerging variants today. This is an area I'm sure that everybody's hearing every day in the news. I know on behalf of Government of Canada, CIHR is very heavily involved in putting together and shaping the response to the emerging variants. So I'm very pleased to provide this update to those who have tuned in today. So it's a series of questions, which I was asked to address and which I thought would be of interest to this audience. So this is a more scientific talk today. So I'm happy to answer any questions, if the science gets too heavy, but I did kind of target it for a scientific audience. So we're going to talk about mutations. We're going to talk about variants of concern. Why are we concerned about this? The possible origins, the clinical relevance, current therapies, and what does that imply in terms of the public health response and vaccine responses. So let's start with what do we mean by emerging variants. So there's thousands of SARS-CoV-2 variants that are circulating and they continue to emerge. I usually have a slide, and for time I didn't put that, but over the course of last 12, 13 months, there have been at least 10, 12 different strains that have emerged, and some of them have died out and some of them have, sort of dominated the world. So for example, the D614 variant, which is shown here, which was initially in the initial part of the pandemic, the dominant strain, is now actually been replaced almost completely with the G614 strain, which is shown in blue on the right side. The changes then can correlate with a number of things, but obviously some of the changes, the reason for the virus becoming dominant can be both founder effects, but also increased transmissibility or increased survival advantage to the virus. The one thing to note is that the SARS-CoV-2 virus are mutating and evolving in a geographically stable manner. That means that they establish themselves, in different regions, and over time they become stable in that area. The point of interest to note here is that global surveillance is ongoing and in fact increasing. And what do we mean by global surveillance? Both increased sequencing, but also epidemiologic survey, which I'll talk about in a minute, the B117, and how that was kind of detected because of both these strategies. And with the combination of these strategies, the earlier detection of emerging virus then allows for localized response and containment. So I'll start off with this slide, which kind of compares the two initial variants that had emerged, the B117 and what was known as the 501YV2, which is now actually B1.351 strain. And we are avoiding using geographical locations to identify viruses. That's a WHO recommendation. So I will be referring to the strain that first emerged in UK as the B117 and the strain that first, or the variant that first emerged in South Africa as the B1.351 strain. Hopefully you guys can remember that terminology. And what I wanted to point out on this slide is that these are unusual variants compared to the ones that emerged over the 12 months or so, because there's an unusually large collection of mutations in there. Some of them in open reading frame and some of them in other regions. But of particular note is the number of variation or number of mutations that was noted in the spike region. So you can see a list of both in the 117 as well as the 501YV2 strain. The number of mutations that are here, which is unusual and much more compared to previous ones. Highlighted in the yellow ones are the ones that we're going to talk about that are of particular interest, because you can have a lot of mutations, but most of them mean nothing in terms of changes in the dynamics of the infection. But these particular ones that are emerging, that have emerged, do have biological implications. So I'm going to talk about that in a couple of slides. But first, I want to share with you, in case you haven't followed this story closely, so that surveillance, the epidemiologic surveillance combined with the genomic surveillance piece. This relates to, how things have evolved with the B117 strain, which is the variant first identified in the UK. And the changes in the clinical epidemiology were in fact the first thing that flagged the reason for doing increased genomic surveillance. And in the southeast of England, you can see in the bottom here sort of the changing graph, which shows the timelines from November to middle of November to late November, followed by beginning of December to middle of December. So it's by every 2 weeks. And you can see over a course of 2 months, the variant spreading from the southeast of England to pretty much now being the dominant strain across the whole of UK. So it's the yeah, 6% to 71% more transmissible. And that equates to an increase in reproductive number to around 0.5 with some variability there. Of course, the reproductive number depends on the shutdowns that are in progress, right? Because it's the number of people you transmit to. The secondary attack rate, which is how many people get infected from, patient zero or each single person and their secondary contacts, is almost double of the original strain of the virus that was in UK. Interestingly, the lockdown that was there in the initial part of November that was there in UK, the B117 was actually showing consistently increased R number, reproductive number, at the same time while the other lineages were contracting. And that was one of the reasons that led to the Public Health England guideline, and they actually went into a stricter lockdown measures than those that were in place since 2020. One of the other interesting parts for this variant is that there's an S gene dropout, which is a 69 to 70 amino acid deletion, which has allowed for very easy detection of this strain, and is now sort of become the proxy marker, which is being used by public health labs all over the world, where you can just do a quick PCR to see if there's a S gene dropout. So those are all interesting facts to note about this, and the fact that this was detected not because of high levels of genomic sequencing, but actually combining the genomic sequencing with unusual transmission and some of the other key factors that were noted. The other thing that I want to flag about the B117 strain is that on the right side, you will see this graph, which shows the age ratio among S negative over the S positive case. So there's a small but significant increase in the B117 prevalence in younger adults, 0 to 19. So you can see that compared to all the other ages, the 0 to 19 ones are higher proportion. So that's a concern, because we know that so far in the previous strains of SARS-CoV-2, the younger people have been less affected than older adults. So this is just to kind of show you that, you know, in the 2 months since this strain was identified, it is now reported in more than 70 countries. And as of 2 days ago, when these slides were made, 324 cases are currently there in Canada. There might have been more last night. I didn't have an opportunity to update that. So that's how quickly these are spreading. A little bit then about the South African or the virus or the variant that first was identified in South Africa, or the B1351, as I'll refer to it. Again, very high number of mutations, which is unusual. And it also has that same type, same N501Y E484 mutations that were seen with the 117 strain. And I'll talk about this a little bit. The E484 mutation is now implicated in immune escape, and was first reported in the context of convalescent serum. The preliminary results also suggest that there's higher viral load for this variant, but so far there's no increase in disease severity. I forgot to mention that the B117, Public Health England is now reporting preliminary data that there may be increased severity as well based on the number of deaths and hospitalizations, but that remains to be confirmed. And then it's now this B1351 strain is now reported in more than 30 countries, including about 18 cases in Canada as of 2 days ago. And then finally, the last strain or last variant that everybody is talking about, which originally was found in Brazil. In fact, the lineage is the B1128 lineage, and there are 2 different variants that are circulating in Brazil. But most of the talk has been on the P1 strain, as it's called, or P1 variant. Which has been detected in Japan first with travelers that went to Japan from Brazil and spread from the Amazonas, Manaus, and North Brazil area. Again, unusually large number of amino acid changes. And again, note that N501Y and E484 and K417T mutations that are now being seen frequently in these mutations. 2 days ago, the first case was detected in near Toronto. So we know that it's here. Again, the implications of how fast it can spread or how severe it is not clear at this point. Okay, so let's talk a little bit about what do these mutations mean and why are they biologically relevant. As I said, not all mutations have biological relevance. So shown in stars are some of the ones that I pointed out are repeatedly showing up in these different variants, whether it's the P1 strain, the 351, or the 117. And many of these, as you will see on the right hand, the impact on viral properties are associated with increased binding to the receptor, increased ability to get into host cells, whether it's animal models or human lung epithelia. You know, some of the deletions, like I said, is important because it allows for diagnostics. Does not appear to have a correlation with immune invasion. It could, but we don't know that yet. And the E484 is the one that's most concerning in terms of the viruses, both from the 351 and the P1, because both of those variants are showing significant immune evasion, not against the vaccines, but in other studies, which I will go over. So just to kind of flag, you know, these mutations and why they are of concern. But before we get into what the implications of that are, I'd just like to take a moment to, you know, talk about the origin of these mutations. Viruses mutate, we know that. The SARS-CoV-2, which is an RNA virus, accumulates mutation once every 2 weeks or so on an average. But most of these disappear and the viruses have no advantage, so there's no replication advantage. So they peter out and don't mean much. But that is a possibility that over time, more and more mutations can accumulate, and then there could be a significant advantage to the virus. But currently, there's no thought that this is how these mutations or these variants have emerged. The immunocompromised individuals hypothesis has gained a bit of traction, more traction, primarily because this has been seen as case reports have shown that in single immunocompromised patients, especially ones who have chronic long periods of RNA shedding for SARS-CoV-2, have been treated with, for example, convalescent plasma or monoclonal antibodies, and still have not recovered, that could potentially push the virus for increased mutation rates or more dramatic mutations. The antigenic evolution, we know that there is a constant tug of war between the host immune system and the viruses. Many of us who work in the HIV area, have known or deal with this, all the time when we are working with these viruses. One of the reasons why we don't have an HIV vaccine yet. And then the adaptation in animal host, which has been shown, if you look back, I didn't have that slide today, but the Ming strain that emerged in Denmark first has shown that the virus can go back and forth between humans and animal host. But which of these really applies to these particular variants, we don't know yet. I'll spend a little bit time over the next 3 or 4 slides really talking about clinical and implications. So first off, many of these variants have been shown to be associated with reinfections now. So there are some case reports and other things that are cited here for B117, also for the P1 and P2 strain in Brazil, where they were shown to reinfect people who had originally recovered from the original SARS-CoV-2 strain and are reinfected. And shown on the bottom is what that could mean on a population basis. So in the Manaus area of Brazil, about 76% of the population had been infected by SARS-CoV-2, the original. And then you can see on the right that after this variant evolved, they are now on sort of on way for a second peak. So kind of like a whole new third wave because of this new variant. So clearly we should be concerned about these variants taking hold, because that could potentially mean that there's a whole new wave because of the changes in the virus. What about therapeutics? So again, a significant amount of data coming out that not the cocktails that are currently approved for therapy, such as the one from Regeneron, which is a cocktail of 2 different antibodies, but single monoclonal antibody treatments, such as the one from Eli Lilly, that can be bypassed by these variants. So both B117, or sorry, B1351 in particular, is of concern because single antibodies do appear to not be able to neutralize it completely. And shown on the right side is a study that's, sort of at the bottom, which actually shows that compared to the old original virus strain and the different antibodies that are shown here, the neutralization, percent neutralization compared to black, which is the old strain, the red and the purple one is significantly lower, showing that the antibodies, single antibodies at least, do not seem to be very good at neutralizing the 351 variant. Also implications for convalescent plasma. I won't get into the detail of it, but there are in vitro studies that are showing that when Viruses are incubated, or the original virus is put together with neutralizing plasma from some of the convalescent plasma patients. You can see that over 90 days, there were 3 different mutations that the virus accumulated, including the 484 mutation, which is of concern. So clearly there is some kind of advantage to the virus, and it likes this, the 501 and the 484 mutation in particular seems to confer some kind of advantage to the virus. And then finally, the vaccine story, which is in the news right now every day. So for the Pfizer and the Moderna vaccine, there's no data. These vaccines were approved prior to the emergence of the variants. So the most studies that have been done are laboratory studies from taking the strains or taking the vaccinated serum and trying to see how good is the serum against these 501 mutations. And for most practical purposes, they seem to be relatively good. The reason that it's thought to be is that it appears to be a very potent polyclonal response. So it's not a single antibody, it's a mixture of antibodies, and that seems to maintain the immune response. However, of note that the cumulative effects of not just the specific mutations, but a combination of mutations may actually have different effect on the neutralization capability. So what I'm showing you here is sort of the summary slides of the vaccines, because I don't have time to go through each one in detail. So we talked about the vaccine, the Moderna and Pfizer vaccine seems to be fairly potent against 117. Against 351, there seems to be some reduction. So I've highlighted in yellow the 2.7 reduction against a single mutation or 3 mutations. But what I was referring to earlier, that if you have the whole set of mutations found in the virus, the reduction in neutralizing capability could be even higher. And then the AstraZeneca, which has been in the news, South Africa just stopped their rollout of the AstraZeneca because you'll see on the right side, the 351 strain against that, there's very low efficacy. However, just to flag that this was a very small study, about 1,750 people divided into placebo and vaccine arm. But it is concerning that there were 20 cases in placebo arm and 19 in the vaccine arm. So really there's not much protection at all, although the confidence interval for the efficacy was pretty high. So again, the takeaway is that we need to do bigger studies. And then Janssen, again, has shown, not as potent efficacy, so somewhat decreased efficacy. And then Novavax, which, was a very nice study that came out, which showed that there's a 60% overall efficacy, which decreased to around 50% if you included HIV positive population. So again, there's issues with the repertoire of antibodies that the individuals are able to produce with the 351 strain. So what are the implications? So clinically, what does that mean when these variants emerge? Like I said, I've discussed examples: higher transmission, severity, increased hospitalizations, deaths, and overall burden on the healthcare system. Vaccines and therapies, we talked about this in the last few slides. Current vaccines could potentially become ineffective. We don't see signs of that yet, but something to be concerned about. And what that means is that we have to have a backup of multivalent vaccines and or second rollout of boosters that we have to get ready for. And then new and cocktails of monoclonal antibodies need to be developed. Public health implications, again, like UK, there may be needs for more restrictive lockdowns if we allow these variants to take a foothold, especially the 117. And age-related transmission. I showed you that the 0 to 19 age may have higher prevalence. And if that turns out to be the case here, then that might affect school reopening. And then the actions, better surveillance and sequencing capabilities. We've talked about that, clinical surveillance combined with genomic surveillance, strengthening national data platforms. So in every country that has successfully identified and is, and is developing a strategy, South Africa, Brazil, UK, they had nice national platforms to basically document what is the implication, clinical, epidemiological, and public health of the variants, and be able to assess what that would mean for other countries as well. And then research to detect and understand early or on the possibility of the potential impact is really, really important in terms of understanding what measures should be put in place. I guess I lost that slide. So there was a slide where I was actually describing the Government of Canada strategy that is being developed, which is actually a combination of both surveillance platform as well as the research arm. From CIHR side, we put out a pre-announcement with a whole three-fold strategy for funding research networks on the variants. And the details of that should be coming out this week in terms of the large network, which will collaborate with the surveillance network. So I'm happy to answer questions at this point. And how did I do with time? Sort of. Okay.
That's okay. It was fantastic. Thank you so much, Dr. Kaushic. But we will get right into the question and answer period. So the first one, I don't see any hands up at the moment, but we have some in the chat box. The first question is, what is the clinical or population health impact of these mutations? And how does this impact of day-to-day lives as well as our health systems?
So I think I covered that. Perhaps this question came in prior to my summary slide. So I kind of summarized that whole list of what the implications are, in terms of clinical implications, public health implications, shutting down, more stricter shutdowns, maybe having to close down schools if the virus is really transmitting at much higher rates in the younger population. Vaccine implications, therapeutic implications. So I went through that whole list. So I think that kind of covers the gamut of the implications.
Absolutely. So the next one is, how are the variants being detected? Sequencing, qPCR. I think you also touched upon that as well. Do you want to?
Yeah, I can comment a little bit about the Canadian strategy. So we know from Government of Canada, the provincial and the federal labs, including the National Microbiology Lab, Canada was doing about 5% sequencing, which is, sounds less, but that's exactly about the amount that UK is doing. So it's actually fairly competitive. There are some countries like, I think Norway, that does about 20%, but most countries, first world countries are in that 5% range. Many are actually lower than that. And currently the strategy is to double that. So the goal is to do about 10% genomic surveillance.
Thank you so much. And the next one is, what percentage of clinical samples are sequenced for surveillance in Canada?
So I just answered that, right? Yeah. So 5%, 10%. I believe that in Ontario in particular, now that the numbers are down, the Ontario Public Health Lab is actually doing the PCR testing for 117, which is the easier test on every sample that's turning positive. And again, there would be about between 5 and 10% complete genomic sequencing.
All right, and the next one is there progress being made on vaccine registry for Canada? We could also be doing more to harness primary care and surveillance. Again, I think you also sort of touched upon that, or?
Yeah, so, this is not my forte. I design funding opportunities and, but I've sat on the tables where the public health data sharing and registries is being discussed. And from everything that I hear, this is a very challenging issue for Canada, given the way we are federated. So I think for sure there is a lot of motivation on behalf of public health laboratories across the country, and the data sharing agreements are being determined. Whether we'll be able to have a single public health registry, like UK. I hope so, but again, this is a public health question.
Absolutely. And then now it's 4:30. We'll just squeeze in one more question because we have lots here. So the next question is, what are the number of serious mutations we can expect in a season/year based on experiences from other epidemics in the past and what we know about this kind of virus? In simple words, will this go on forever?
Yeah, so I don't think we can take what happens in influenza or HIV to coronavirus. They're all different viruses. And like I said, coronaviruses are kind of like somewhere in between very stable viruses like DNA viruses like herpes compared to HIV, which mutates in every person and, very high rates. So it's somewhere in between. Normally it accumulates 1 to 2 mutations a month. But we know that, like I said, it's not clear how it accumulated 23, 24 mutations, in these variants that are showing up. It remains to be seen whether this level of mutations and variants are occurring everywhere. And is the virus gaining some kind of advantage or learning after 13, 14 months? The basic public health message remains the same. And I think you've heard Dr. Tam, Dr. Fauci say the same thing. If we can control the replication, the virus cannot mutate. So I think while we wait for vaccine rollout, probably the most important thing that we can do is to control the replication of the virus so that it doesn't have more chances to mutate.
Great. Thank you so much, Dr. Kaushic. We do have lots more questions in the chat box. Unfortunately, though, we are out of time, but I just want to take this opportunity to thank you so much for presenting to us on this important and topic that people have lots of questions about because it's, it's in the news and everything. So thank you so much again. And I just want to announce that I hope we see all of you next week at our next speaker series on Tuesday at 4 o'clock next week. Thank you so much.
Thanks very much for the opportunity. Take care.
Bye-bye.